everyone, we are come from Johor Matriculation College. So now we are going to talk about the topic chapter 5, cellular respiration and fermentation. We will start with the subtopic glycolysis. So actually what is glycolysis? Glycolysis is the breakdown of glucose. So we know that glucose is the most important carbohydrate which is used as the substrate for the oxidation. So what is the use for oxidation? Is because this is the cellular respiration which can provide us enough energy to conduct our daily life. So this is glucose C6H12O6 and it undergoes oxidation and it will release energy. So in the form of ATP. Okay, so this is glycolysis. So glycolysis is about the breakdown of glucose and the product is energy and also pyruvate. So glucose is the 6 carbon and sugar and is split into 2 molecules. So if 6, 1, 2, 3, 4, 5, 6, so it's split into half. So this is 1 pyruvate, this is the number 2 pyruvate. So 1 glucose will produce 2 pyruvates. Okay, so it is split into 2 molecules of pyruvate. So the pyruvate is 3 carbon sugar. Okay, and the glycolysis can be occurs in both aerobic and also anaerobic respiration. Aerobic means with oxygen and anaerobic, this N means without or no. No oxygen means without oxygen. So it occurs in cell cytoplasm or cytosol. Okay, glycolysis, the main key we need focus is the important products are 2 ATP nets and 2 NADH. So it contains 2 phases, which is energy investment phase and energy payoff phase. What is energy investment phase? Which means before we can produce our product, we need to invest the ATP first. So after we invest the ATP, we only can get back the ATP. So at first, we need to invest 2 ATP. Okay, we need to invest 2 ATP first. This is a rough idea. We need to give up 2 ATP and finally we will gain 4 ATP. So actually 4 minus 2 because 2 we already used before. So our products are 2 ATP net. Okay, because although we gain 4, but at first we already used 2. So our product is 2 ATP net. Okay. So the energy investment is the cell spent ATP and the energy payoff phase means that by substrate level phosphorylation, the NAD plus is reduced to NADH and electrons released from the oxidation of glucose and the energy is repaid when the ATP is produced. Okay, so actually what is substrate level phosphorylation? Substrate level phosphorylation means we gain the phosphate group from the compound which is phosphorylated. Okay, later we will see this. So at first the glucose, okay, so at first our reactant is glucose. Okay, glucose is monosaccharide, means that normally what we eat and then finally it will become the glucose. Okay, and then it catalyzed by the enzyme hexokinase, it will produce glucose 6-phosphate. So see the 6 carbon before this, it doesn't have phosphate, but after catalyzed, there is 1 phosphate group above 6 carbon. So it's called 6-phosphate. Okay, this is why the name is called glucose 6-phosphate. And the ATP here, it already spent 1 ATP. Okay, so he spent 1 ATP. So ATP becomes ADP and P. Oh, sorry. P. So here the P moves to this carbon. Okay, and the ADP is here. So we already spent 1 ATP. And they undergoes rearrangement and form fructose 6-phosphate. Okay, fructose 6-phosphate. It undergoes isomerization. And the fructose 6-phosphate, it will undergoes phosphorylation, which catalyzed by phosphofructokinase. Phosphofructokinase, it catalyzes the 6-phosphate to the 1,6-biphosphate. So it means that the fructose 6-phosphate gain 1 more phosphate group on the first carbon. And the ATP, it actually is same. ATP will become ADP plus P. So this P move to here. So fructose has 2 carbon. Okay, now fructose 1,6-biphosphate, it will split. Okay, split. Okay, you imagine they already have 2 phosphates, so they already become rich. And now when they're rich, they split up, and this is formed G3P. And here, it is DHAP. What is the difference? G3P, the phosphate group is on the right, but the DHAP, the phosphate group is on the left. So DHAP, it needs to rearrange in order to become G3P. So now we already have one G3P, and the DHAP is rearranged to become G3P. So we have another G3P. So total we have 2 G3P. Okay, and the G3P, they will undergo oxidation and phosphorylation. So the G3P undergoes oxidation. What is oxidation? It means they throw out the hydrogen atom, right? So actually, each G3P that transfer 2 hydrogen atom, and the hydrogen atom makes the NAD plus become NADH and also H plus. So if one G3P transfer 2 hydrogen atoms, so 2 G3P that transfer 4 hydrogen atoms. So 2, 2, okay, 4. Okay, so each G3P, remember, that transfer 2 hydrogen atoms. So transfer hydrogen means undergoes oxidation. And phosphorylation means G3P, the first carbon here, they gain phosphate group again. So the name is 1,3-biphosphoglycerate. Okay, for 1,3-biphosphoglycerate, now we already see they have 2 phosphate groups. So means they are rich, right? Just now they already borrow the ATP here. So 1 and 2. We already spent 2 ATP. But now they have 2 phosphate group, means they are rich. So for example, like you borrow the money from others, and now you already have money, so you need to pay back. Okay, so here, because 2 biphosphoglycerate, so each of them, they donate the phosphate group from the first carbon, and there are 2 phosphate group being thrown out. So 2 ADP plus, each of them plus P. So I can write here 2 because 2 ADP, right? So it will produce 2 ATP. Okay, so here we already pay off because just now we spent 2 ATP. Now we gain 2 ATP. So we already pay back the 2 ATP. And now the product is 3-phosphoglycerate. Okay, and then from this is compound which is already phosphorylated. 1,3-biphosphoglycerate is the compound which is phosphorylated, and he donates the phosphate from itself and from ADP to form ATP. So this process called substrate level phosphorylation. Okay, means from the phosphorylated compound, they donate the phosphate group or transfer the phosphate group, and from ADP to produce ATP. This process is called substrate level phosphorylation. So form 3-phosphoglycerate, and rearrangement form 2-phosphoglycerate. And here there are removal of water molecule, and so 2-phosphoglycerate, they will become phosphoenolpyruvate, or we can call PEP. This is the short form PEP, and the PEP here has the phosphate group, right? So they donate out the phosphate and become pyruvate. So if each of the phosphoenolpyruvate, they throw out 1 phosphate, so total we have 2 phosphate. So 2 ADP plus 2P, it will form 2 ATP. And the concept is totally same as above. So this is the process of substrate level phosphorylation, which the phosphate group comes from the phosphorylated compound and makes the ADP becomes ATP. So our final product is 2 pyruvate, and also we get extra 2 ATP and also the 2 NADH, which comes from here. So 2 NADH and also 2 ADP and also 2 pyruvate is our final product. That's all for glycolysis. Thank you.
Hello everyone, my name is Nadh Dehydrogenase. I want to present about the LING reaction and Krebs cycle. First, we begin with the LING reaction. Okay, LING reaction occur in the matrix of mitochondria. Okay, the LING reaction occur need oxygen. Oxygen is needed for LING reaction, but in the glycolysis, oxygen is not needed. So it is the difference between the LING reaction and the glycolysis. In the LING reaction, the process that occur is called oxidative decarboxylation. So why it is called oxidative decarboxylation? Because it is the combination of oxidation and decarboxylation. The first product of the LING reaction is pyruvate. Pyruvate is a 3C compound. The pyruvate is come from the final product of the glycolysis. Okay, at first, oxidation. The pyruvate release the 2 hydrogen ion, and this hydrogen ion is accepted by the NAD plus to produce NADH and H plus. After the oxidation, the decarboxylation occurs. The carbon dioxide is released, and the core enzyme is accepted by the pyruvate. After these three processes, the pyruvate will become acetyl-CoA. Okay, so this is the Link reaction. So the Link reaction is when pyruvate becomes acetyl-CoA. After the Link reaction is the Krebs cycle. Okay. Krebs cycle also occurs in the matrix of mitochondria, and oxygen is also needed for Krebs cycle. Okay, matrix of mitochondria is at here. Okay. Then we begin with the first step of the Krebs cycle. The first product of the Krebs cycle is acetyl-CoA. The acetyl-CoA is come from the Link reaction just now. The acetyl-CoA react with the oxaloacetate, release core enzyme and produce citrate. Then the next step is the citrate undergo isomerization. So what is isomerization? Isomerization is the rearrangement of compound. In order to undergo the isomerization, the citrate need to accept the water, and the water is released to form isocitrate. Okay, the third step is isocitrate become alpha-ketoglutarate. At here, the process that occurs is called Oxidative Decarboxylation. It is also the combination of oxidation and decarboxylation. The isocitrate undergo oxidation because isocitrate release 2 hydrogen ions. And this hydrogen ion is accepted by the NAD+, and NAD+ turn to NADH. Meanwhile, the isocitrate also release carbon dioxide. So, after the oxidation and decarboxylation, the isocitrate become alpha-ketoglutarate. Then, the fourth step is also called oxidative decarboxylation. The alpha-ketoglutarate undergo oxidation and release 2 hydrogen ions. And this hydrogen ion accepted by the NAD+ and produce NADH and H+. Meanwhile, the alpha-ketoglutarate also release carbon dioxide. But at this time, the alpha-ketoglutarate accept the core enzyme. So the product will have the core enzyme at here. This blue circle is the core enzyme. So after oxidation, decarboxylation, and alpha-ketoglutarate accept the core enzyme, it will form succinyl-CoA. And when the alpha-ketoglutarate accept the core enzyme, the succinyl-CoA become high energy and the bond between them is unstable. So let us proceed with the fifth step. Okay. Then the succinyl-CoA will release the core enzyme. When he releases the core enzyme, the energy is released. The energy is then accepted by the GDP. Okay? The energy is released in the form of phosphate. Okay? The phosphate is accepted by the GDP, and GDP turn to GTP. But the GTP doesn't like the phosphate, so he donates his phosphate to the ADP. So after ADP accepts the phosphate, it's become ATP. This is the site where the ATP production, and we call substrate level phosphorylation. And in the next step, the succinate undergo oxidation. The 2 hydrogen ion is released, and the hydrogen ion is accepted by the FAD to produce FADH2, and succinate turn into fumarate. Then the fumarate accept the water molecule and it turn to malate. After that, malate undergo oxidation because he released 2 hydrogen ions. And the hydrogen ion is accepted by the NAD+ and it's produced NADH and H+. And then the final product of the Krebs cycle is called oxaloacetate. After produce the oxaloacetate is then used again by the acetyl-CoA and the cycle, and then we go to the next Krebs cycle. So the process is go around. Okay. In conclusion, okay, oxaloacetate is regenerated and bind with the acetyl-CoA, so the cycle is continuous and repeated. In conclusion, the Krebs cycle produces 6 NADH, 2 FADH2, 2 ATP, 2 CO2. Okay, just now because we got 2 acetyl-CoA, so the product that produced just now must multiply with 2. So we produce 6 NADH2, FADH2, ATP, and 2 CO2. Let me teach you to remember the sequence of the Krebs cycle because it's difficult, right, to memorize the chemical name. We can use the happy birthday song to memorize this process. Okay, let me perform. Okay, We can memorize like citraisocetra, alpha-ketoglutarate, succinate-CoO, succinate, fumarate, malate, oxalo, etc. Okay, so we can use this music to memorize this Krebs cycle, and it's very easier for us to memorize it. Okay, that's all. Thank you.
Good morning to everyone. Today I will present about oxidative phosphorylation, and I will focus at electron transport change and chemiosmosis. What is oxidative phosphorylation? Alright, oxidative phosphorylation is a couple of mechanisms to synthesize ATP by phosphorylation of ADP using energy derived from transfer of electron in ETC and chemiosmosis process. So, what is ETC and chemiosmosis? Alright, so now this is the ETC, and this is the inner membrane of mitochondria. This is the intermembrane space in mitochondria, and this is the mitochondrial matrix. The first enzyme is Nadh dehydrogenase. The second is ubiquinone, or also known as coenzyme Q. This is succinate dehydrogenase, and this one is cytochrome C reductase. This one is cytochrome C. And the last one is cytochrome C oxidase. This is for ETC, and for chemiosmosis, this is ATP synthase. And now I will explain how the electron pass through the ETC. Alright, first the NADH will pass electron to NADH dehydrogenase because the NADH dehydrogenase is more electronegative than NADH. And when the electron is passed to this enzyme, there's an energy release, and the energy is used to pump proton from the matrix to intermembrane space. Yes, which the H plus pump to intermembrane space. And after that, the electron will pass to coenzyme Q, and next it passes to cytochrome C reductase. And here, there's also energy release, and the energy is also used to pump H plus from matrix to intermembrane space. Next, the electron will pass to cytochrome C and pass to cytochrome C oxidase. And the same as before, the energy release is also used to pump H plus from matrix to intermembrane space. And finally, the electron here will bind to the oxygen and also bind to the H plus here to form H2O, which is the water. And for FADH2, this is different from NADH. The FADH2 is more electronegative than the NADH dehydrogenase, so it cannot pass electron to NADH dehydrogenase, but it passes electron to succinate dehydrogenase. And after that, the process is same as the NADH, which pass to coenzyme Q, pass to cytochrome C, and pump proton, and pass to cytochrome C, and pass to cytochrome C oxidase and pump proton, and the last, the electron accepted by the oxygen. Alright, so this is the process of ETC. And after ETC, there's many H plus which the proton accumulate in intermembrane space. The concentration of H plus in intermembrane space increase, but how is the H plus returned back to the matrix? The H plus need to diffuse through this ATP synthase, since it cannot just pass through this membrane layer. No, it can only diffuse through ATP synthase. And when the H plus diffuse through this enzyme, the energy is released. Then the energy is used to bind ADP and a phosphate group to become ATP. So from here, ATP is produced by the chemiosmosis. Yes, this process is what we call chemiosmosis. For one NADH can pump 3 H plus, but one FADH2 can only pump 2 H plus. Yes, do you remember that before this? Because the FADH2 is more electronegative than NADH dehydrogenase, so the NADH2 will pass to succinate dehydrogenase. But succinate dehydrogenase do not pump H plus from matrix to intermembrane space. So for NADH, it pumps 3 times. Here, this is the first time, the second time, and third time here. But for NADH2, it only pumps 2 times, which this is the first time, and this is the second time. So we can conclude that one NADH only pumps 3 H plus, and only 3 HTP is formed. And one FADH2 can pump 2 H plus, and only 2 ATP is formed. Okay, so after all, we can conclude that one oxidative phosphorylation can form 34 ATP because there's 2 NADH from glycolysis, 2 NADH from link reaction, 6 NADH from Krebs cycle, and 2 FADH2 from Krebs cycle. So we times NADH times 3, FADH2 times 2, and we can get this the ATP. And finally, we get 34 ATP. Alright, so that's all for oxidative phosphorylation. Thank you.
Hello everyone, I would like to talk about the ATP production and fermentation. Before that, I want to introduce the two ways of ATP production, which is the substrate level phosphorylation and oxidative phosphorylation. So what is the substrate level phosphorylation? It is directly ADP will turn into the ATP when you receive a group of phosphate. So the second way is the oxidative phosphorylation, which is occur in the ETC. In the ETC, one NADH will produce 3 ATP and one FADH2 will produce 2 ATP. Okay, so that's the way of ATP production. So let us proceed to the summary of the aerobic respiration. So now we would like to talk about the ATP production in the aerobic respiration. ATP production. So first we look into the process glycolysis. In the process glycolysis, 2 ATP is produced in the substrate level phosphorylation. This is from the substrate level phosphorylation. In the link reaction, there is no ATP produced, while in the Krebs cycle, there are 2 ATP produced also through the substrate level phosphorylation. As you know, the substrate level phosphorylation is one of the way of the ATP production. So the second way is the oxidative phosphorylation. In the glycolysis process, 2 NADH is produced, so they will produce 6 ATP. In the link reaction, there are 2 NADH produced, so they will produce and also 6 ATP. In the Krebs cycle, 6 NADH is produced, so and also they will produce 6 times 3 total 18 ATP, while the 2 FADH2 will produce 2 times 2 total 4 ATP. Yes. All of this is the oxidative phosphorylation. So from here we can compute that there are total 38 ATP is produced in aerobic respiration. However, there are a range that 36 to 38 ATP produced. ATP is the maximum ATP produced in the aerobic respiration. So the range of ATP produced is actually depends on the glycolysis that we call that shutter. There are 2 types of shutter, which are maleic shutter and also glycerol 3-phosphate. The maleic shutter will produce 38 ATP, while the glycerol 3-phosphate will produce 36 ATP. So there will be a difference in the total ATP produced in the aerobic respiration. So that is the summary of the ATP production in aerobic respiration. Now we would like to talk about fermentation. So actually, what is the fermentation? Fermentation occurs in the condition absent of the oxygen. After the glycolysis, if there are absent of oxygen, we will undergo the fermentation, while if the oxygen is available, we will undergo the link reaction. There are 2 types of fermentation, which are lactate fermentation and also the alcohol fermentation. The lactate fermentation is occur in the animal, while the alcohol fermentation is occur in plants or yeast. The fermentation is occur in cytosol because without the oxygen, the pyruvate from the glycolysis cannot enter the mitochondria. So it will stay in the cytosol and undergo the fermentation. So why we need to undergo the fermentation? Actually, it is needed for us to sustain the glycolysis process. In the glycolysis process, we need a lot of NAD+ as a reducing agent. So when the glycolysis is occur and the pyruvate cannot enter the mitochondria to undergo the further oxidation, there will be an NAD+ shortage. So we need to undergo the fermentation to gain more NAD+. So the glycolysis will continue. Now we will talk about the first fermentation, which is lactate fermentation. Okay, the fermentation is also same with the aerobic respiration, which it also undergo glycolysis.
So as we know, in the glycolysis, the glucose is oxidized to 2 pyruvate, and also it will produce 2 ATP from ADP. So at the same time, 2 NAD plus will reduce to NADH and hydrogen. So when 2 NAD plus will reduce to NADH and H plus, 2 pyruvate will receive the electron from the NAD plus and reduce to lactate, and NADH will oxidize to NAD plus. So the total ATP produced in the lactate fermentation is only 2 ATP, which is already produced in the glycolysis. The second fermentation is alcohol fermentation, which occur in the plants or bacteria and yeast. Yeah, so same as the lactate fermentation, it also undergo glycolysis, which glucose will oxidize into 2 pyruvate, and also 2 ADP will receive a group of phosphate and turn into 2 ATP. Same as the NAD plus will receive the electron and turn to NADH plus and 2H plus. So there's a difference in this alcohol fermentation compared to the lactate fermentation, which the pyruvate will not directly receive the electron from the NADH plus. It will undergo the decarboxylation, which it will remove a carbon dioxide and turn into 2 acetaldehyde. So the acetaldehyde will receive the electron from the 2 NADH plus and reduce into ethanol. So when the NADH plus contribute the electron to the 2 acetaldehyde, it will also be oxidized into NAD plus. So the NAD plus is produced in the fermentation, which can sustain the glycolysis process. Same as the lactate fermentation, alcohol fermentation also produce 2 ATP, it's a total ATP. So as a summary, the product from the lactate fermentation is 2 ATP and lactate, while the product produced in the alcohol fermentation is 2 ATP and also a carbon dioxide and ethanol. Okay, so that's the process of fermentation. Yeah, so that's all from me. Thank you.